Hi, Assalamualaikum Dr. Hanis. My name is Nabila Huda. My group presentation is about improvements of sugarcane for borer resistance using Agrobacterium mediated transformation of Cry1AC gene. Let's begin with introduction. As we know, sugarcane is the major supply of sugar in the world. However, insect pests are an essential problem for sugarcane crop. One of the effective strategies to overcome this problem is by introducing the insect resistance gene, which is Cry1AC gene. Recently, many monocotyledonous species use Agrobacterium transformation method as it have diverse advantage. Thus, the purpose of this study is to improve the borer resistance in sugarcane plants via introducing Cry1AC gene through Agrobacterium transformation. Further explanation will be continued in the next slide.
Assalamualaikum, my name is Nur Farah Hanim and I will continue presenting on Agrobacterium transformation. Recently, the researchers conduct a study to produce sugarcane that are resistant to insects, such as borer. Hence, they use sugarcane from cultivar GT54-9C9. First, they co-cultivated young explants with Agrobacterium tumefaciens GV3101. And then after 3 days, the inoculated explants were transferred to MS medium and kept in dock at 28 degrees Celsius. 48 hours later, the explants were taken out and washed with strength MS medium and carbonicillin solution. Then, they blotted it on filter paper inside the laminar flow hood. After that, the explants were transferred to regeneration media that was supplemented with kanamycin and carbonicillin. The explants then were incubated for 30 days so that they can subculture the shoots to fresh medium with same antibiotic and re-incubated under same conditions. Next, they transferred the regenerated shoots to medium supplemented with sucrose and NAA to induce the roots. Finally, the regenerated plants will be transferred to pots and kept in greenhouse for acclimatization. For further analysis, researchers will take 16 transgenic sugarcane to develop and grow on MS media added with kanamycin. ISSR marker were used to analyze the result, and it was reported that there are no phenotypic abnormalities appeared on transgenic sugarcane. And that's all from me. Thank you.
The researchers had been used two methods for detection of transgenic sugarcane, which are PCR and southern blot. So firstly, I will explain about detection of transgenic sugarcane by using PCR method. The transformed kanamycin-resistant sugarcane shoots were used for DNA isolation. The PCR was used to confirm the Cry1AC gene integration into genetic material of the transgenic sugarcane cultivar GT549C9 using NPT, 2-Roman and Cry1AC specific primers. The specific primers were designed to amplify fragments of the NPT2-Roman and Cry1AC genes. Only 20 out of 90 sugarcane plants give positive results, which are the PCR tests show a clear band corresponding to the relevant sequence of both primers. The positive result shows the transgenic sugarcane are present. Detection of transgenic sugarcane by southern blot method. Southern blot was commonly used to confirm the integration of the Cry1AC gene in the T1 plasmid sugarcane plant. The restriction enzyme KPNI was used to digest sugarcane genome, and then digested DNA was hybridized with Cry1AC specific probe that showed the integration of Cry1AC gene in sugarcane genetic material. Linear PRT Cry1AC plasmid DNA was used as positive control. Southern blotting test for transgenic sugarcane showed band more than 3,256 base pairs molecular weight.
for the expression of Cry1AC gene in transgenic sugarcane plant, the stable expression of the Cry1AC gene in the transgenic sugarcane line was confirmed by using reverse transcription polymerase chain reaction, or we call as RT-PCR. The total RNA was extracted from PCR positive putative transgenic line and also from the non-transgenic plant, which act as negative control. Then, the extracted RNA was used as a template in RT-PCR for synthesizing the cDNA, followed by the amplification of the Cry1AC gene with Cry1AC specific primer. Next, based on the figure, the results showed that the RT-PCR fragment with molecular size about 497 base pair was amplified from total RNA isolated from transformed plant. The RT-PCR analysis for the sugarcane plant showed the occurrence of the mRNA for the Cry1AC gene in 6 out of 8, which also count as 75% PCR positive plant for sugarcane.
In order to detect the presence of Cry1AC protein in transgenic sugarcane lines, the study proceeded to the survey with trip test for the Cry1AC protein. According to the study, there are several materials and methods that included in this experiment. Quick sticks combo kit for Cry1A and Cry2A were used to detect Cry1A protein in transgenic sugarcane lines. 2 leaves were taken from each plant and punched by snapping cap of a Pandolf tube. Then, they were grounded by rotating pastel until fine grinding. The extraction buffer with 0.5 ml was added to the tube and mixed with the leaf tissue. Quick stick strips were dipped in the leaf and examined for 10 minutes. The final bands on strip were present and results were recorded. The results and observation. Based on the picture, there are 8 transgenic sugarcane lines that are tested by the quick stick test to detect the presence of Cry1AC protein. In the strip containing lines 6 and 8, only the assay band was observed. This indicated that the absence of Cry proteins. In sample containing the Cry1AC, which is lines 1, 2, 3, 4, 5, and 7, both assay and expression band were detected. This indicated that those lines containing the Cry1AC protein.
Assalamualaikum warahmatullahi wabarakatuh. My name is Nur Aina Mardia with metric number 1171500, and I will explain on the result of the bioassay method. Okay, first of all, bioassay method is an important method because from this method we can evaluate the potential of the insect-resistant transgenic plant against the target pest. Okay, so this is the result. The table shows the mortality percentage of Sesamia critica, which is one of the Lepidopteran insect pests that cause problem to the sugarcane crop. And as you can see, there were 6 transformed sugarcane had been selected against 1 non-transformed sugarcane. The lethal concentration 50 means that the concentration at which half of the insect die, and this LC50 value for Cry1AC toxin protein is at 300 ppm and also 500 ppm. Apart from that, at 1000 ppm, the percentage of mortality of 6 transformed sugarcane is 100% compared to the non-transformed sugarcane. To highlight, this means that the expression of Cry1AC gene in the transformed sugarcane is high. Lastly, this is the comparison between the transformed sugarcane that reach 100% mortality at 1000 ppm and with the non-transformed sugarcane. So that's all from me. Thank you.
Transgenic plants expressing Cry for methylphalangenesis will minimize the use of genetic insecticide and more effective in controlling pests than methylphalange formulation. In 1995, the Environmental Protection Agency (EPA) in United States approved the commercial production and distribution of Bacillus thuringiensis crops, including corn, cotton, and tobacco. The examples of the available of commercial production and distribution crops are Bacillus thuringiensis soybean, varieties expressing the cry1AC gene in Latin America, and also the Bacillus thuringiensis sugarcane, CTC175 for commercial use in Brazil. Cry from Bacillus thuringiensis. A large number of strategies has been tested and described to against the main insect pest. Many researchers suggested that toxicity from the cryogens is due to the poor formation ability and signal translation pathway by receptor binding. The mode action of model of Bacillus genes suggests the cryotoxin pass through a specific binding mechanism with several insect gut proteins leading to membrane insertion, pore formation, and toxin oligomerization. In Bacillus bacterial cell, the cryotoxin produced is called as protoxin. A high yield of Bacillus cryotoxin could be produced by insect gut protease enzyme. Both cry1A protoxin and the activated toxin binds to cadherin receptor forming link-state oligomers that enter into the membrane forming lytic pores. The outcome will be the decreasing of visibility, aberration of resistance, and possibility to expand the spectrum of insect targets. Agrobacterium transformation method. Cry1AC gene was the first reported in cotton for insect resistant to bollworm in the scientific name Helicobacter armigera, and it is also a reliable method and efficient in sugarcane. Agrobacterium transformation method has traditional method to generate with low transgene copy number. In this research, sugarcane variety GT549 was transformed with Agrobacterium tumefaciens GV3101 that have the cry1AC binary vector. The stable integration of the cry1AC gene into sugarcane DNA was assured by using the PCR and southern blot analysis. Southern blot analysis is considered the traditional method to assess copy number of exogenous genes in transgenic plants. The southern results show the gene copy number was integrated in 1 to 3 positions in sugarcane plants that were transformed using Agrobacterium method. Quiz state was used to quantification determination of proteins. And RT-PCR, which is the real-time PCR, used in transformed cry1AC sugarcane plants to reveal the expression of gene and detected that all plants express mRNA of the transgene.
Next, the second part of the discussion is the simple effect of cry1AC expression when tested for the death rate of Caesamea pratica when fed to transgenic PT sugarcane. These findings revealed that a significant amount of PT protein present in all transgenic lines of the sugarcane and the cry1AC gene could be used for target Lepidopteran insect pest control. The mortality rate of cry1AC toxin expressed in all transgenic plants against Caesamea pratica was 100% with 1000 ppm compared to negative regulations. These results showed a high expression of the cry1AC gene in the refined sugarcane plant. The findings revealed that sugarcane line 8 showed the highest toxicity to larvae at low concentration, followed by the line 6A, which could be attributed to the incorporation of just one code of the gene as a result of the southern blot analysis. Many former researchers find that multiple cDNA inserts display less expression than single copy transgenes. These findings are also similar to Lin et al. Finding that the bioassay with cry1AC transformed transgenic tobacco plants showed a high degree of toxicity to Spodoptera littoral with a larva mortality rate of 76.9 to 100% after 72 hours. Earlier, the sugarcane cultivars COG 64 and CO 86 Cry132 had been modified by the Cry1AB gene. The percentage of Cry1AB protein in many transgenic lines range from 0.007 to 1.73% for total soluble protein in leaves. At the seedling stage, transgenic plants were substantially less dead, and there was a negative relationship between dead and protein expression. When et al. Found that only resistance was shown in transgenic sugarcane lines expressing Cry1AC protein more than 9 nanogram or milligram when the pest resistance of Cry1AC was analyzed. There was also a positive association between pest resistance and Cry1AC material.
Okay, Assalamualaikum. My name is Muhammad Amir Nazhan bin Muhammad Yusof. My matric number is 1171508. I will present about the conclusion. Okay, first, the transgenic sugarcane with Cry1AC gene that has been inserted into sugarcane genetic material by Agrobacterium transformation method. It showed resistance to insects and high productivity to the sugarcane. From different molecular analyses, confirm that crystal protein gene is stable, integrated into transgenic sugarcane genome. Okay, that's all from our group. Thank you.